Plasma membrane. Cell membrane is also called as plasma membrane, outermost covering of the cell that separates the contents of the cell from its external environment. Plasma membrane is flexible and is made up of organic molecules called phospholipids, which are phosphate head and lipid tail and proteins. Cell membrane is made up of phospholipids and protein. The flexibility of the cell membrane enables the cell to engulf food and other material from its external environment. Such processes are known as endocytosis. As shown here, amoeba get food through the process of endocytosis. The plasma membrane allows or permits the entry and exit of some materials in and out of the cell through a process called diffusion. Diffusion is the net movement of molecules or atoms from a region of higher concentration to a region of lower concentration. Here we can see process diffusion of carbon dioxide from inside the cell to outside the cell, as the concentration of carbon dioxide is higher on the inside of the cell. Carbon dioxide passes through the plasma membrane. Even when the concentration of oxygen is higher on the outside of the cell, the oxygen passes through the plasma membrane to the inside of the cell. Once the concentration between the inside and outside of the cell becomes equal, then the diffusion stops. If water, which is a solvent, moves through semi-permeable membrane into a region of higher solute concentration, like salt, it is called osmosis. Osmosis is the net movement of solvent into a region of high solute concentration through a semi-permeable membrane. Types of osmotic solution. A hypotonic solution occurs when the concentration of the solute outside the cell is less than the concentration of solute inside the cell. Since concentration of water is more outside, water enters the cell and the cell swells. The osmotic pressure outside is more. An isotonic solution is one that has the same solute concentration in cell as another solution. Since osmotic pressure is equal, there is no change in the cell. A hypertonic solution is a solution that has a greater concentration of solutes on the outside of a cell when compared with the inside of a cell. As water concentration is more in cell, water moves outside and thus cell shrinks. Osmotic pressure inside the wall is more in cell than the surrounding. Please note that concentration of solution is equal to the amount of solute divided by the amount of solvent. Let us perform an activity with eggs and raisin. Add some raisins in a beaker containing distilled water, and likewise add some raisins in another beaker containing salt water. Leave them for a while. We will repeat the same activity with egg, removing its shell. What do we observe? We observe that both raisins and egg in distilled water swells, while the one in salt water shrinks. This happened as it is based on the difference in osmotic pressure. Plasmolysis is the process in which cells lose water in a hypotonic solution. Unicellular freshwater organisms and most plant cells tend to gain water through osmosis. Absorption of water by plant roots is also an example of osmosis. Let us take a glass slide with a drop of water on it. Gently scrape the inside surface of the cheek using an ice cream spoon. With the help of a needle, transfer the material that was stuck on spoon and spread it evenly on the glass slide. To color the material, we can put a drop of methylene blue solution and put a cover slip on it. Now observe this glass slide under microscope. We will observe darkly colored spherical or oval, dot-like structure near the center of each cell. Cell wall absent, a distinct nucleus, lightly stained cytoplasm in each cell. No prominent vacuoles. This structure is called nucleus. We saw similar structures in onion peel cells too.